All right, hello everybody. We're going to go ahead and get started today. We're going to be continuing with meiosis and talking about how meiosis can lead to genetic variation in organism populations and why that is a big deal as it applies to things like natural selection. So today we talked a little bit about how meiosis works. Here's a quick rundown in which we have a diploid cell. DNA is replicated in interphase, goes through meiosis 1, which we see here, and then later on goes through meiosis 2, which we see here, and we end up with 4 haploid cells. Now today in class I had talked about this idea that sometimes genetic information can be exchanged during the meiotic process, and that's what we're going to talk about today. Now what can happen is when you have these double chromosomes, which we have here, so we still have homologous chromosomes here and here and here and here. What can happen during prophase 1, so during the first round of meiosis, is these homologous chromosomes can exchange information at different locations. So, for example, if we look here, over here, this is the product of meiosis 1, we can see that genetic information was transferred at this location and this location from here to here. So we can see some dark blue and some light blue. So what we see is genetic information was literally exchanged between homologous chromosomes. And we refer to this as crossing over. Please remember that crossing over occurs during prophase 1. So today we're going to talk about why that's a big deal and how that creates genetic variation. So let's look at a different example here. And let's look at what happens when we don't have crossing over. So we have a bird population here, and we're going to pretend that this bird population has 2 homologous chromosomes. Here is set 1, and here is set 2. Okay. Now, the chromosomes go through meiosis. When it's all done, these are our 4 haploid cells. Now we can see that we have 2 traits. We have trait A, we have trait B. And trait A and B are going to dictate a couple different traits on the bird. Now, if this parent were to make offspring haploid cells, the only combination that it can make is lowercase a and uppercase B, or uppercase A and lowercase b. So when the haploid cells are done splitting, right, meiosis 1 would split it here, and meiosis 2 would split it here. These are the 4 possible haploid cells that we can have. But in actuality, there's only 2 possible haploid cells here, little a, big B, big A, little b. So these are the 2 possible offsprings that the organism could have. Now, how would this change with crossing over? Well, let's say that crossing over does exist. And what happens is this B allele, or this B trait, is exchanged between the homologous chromosomes. And then we go through meiosis 1 and meiosis 2, and what we can see here is that our haploid cells, or our possible haploid cells, have changed. Now we can have little a and big B, little a, little b, big A, little b, and big A, big B. So what we've done is through this crossing over, we have increased the amount of different genotypes or phenotypes that we can possibly have in our population. So crossing over in this specific example doubled the amount of genetic variation that we have because we are allowed to exchange information amongst our chromosomes. Now, besides crossing over, there's also going to be another way that we can increase genetic variation, and this is through something called random assortment. And what this means is that when chromosomes line up, they don't always have to line up in the same way. So if we go back to our previous example here that we talked about in the first part, every time I showed these homologous chromosomes, I always had the dark chromosome with the light blue here, and then the light green here, and the dark green here. And I never varied from this in all my examples. In actuality, that's not always how it works. Where these chromosomes line up during the metaphase is completely random. So our diploid cell could have dark blue over light green, or it could have dark blue over dark green. So they don't always line up in the same exact way. The way they line up is completely different. And we refer to this as random assortment. And this random assortment is going to increase the amount of variation that we have in our population. So we can actually look at an example here. So here is my regular diploid cell without random assortment. And if I were to go through meiosis 1 and meiosis 2, the only possible haploid cells I can make is light colored and blue with dark colored green, and dark blue with light green. These are my only two possible haploid cells. However, with random assortment, I can switch that the dark blue is lined up with dark green. I'm sorry, guys, that's my cat. Oh, go. We can have dark blue with light green or dark blue with dark green. So the amount of different variations that we can have increases, because now I can have light blue with dark green, dark blue with dark green, dark blue with light green, or light blue with light green. So the amount of different haploid cells that we can create has now doubled thanks to random assortment. Now, the last source of genetic variation for haploid cell is going to be fertilization. What this means is which sperm lines up with what egg is going to be completely different. So this top sperm can line up with any of these 4 haploid cells. This one can line up with any of these, or any of these. So there's not going to be a fixed genetic encounter. And the randomization of what haploid cell lines up with what other haploid cell from the other parent is going to increase our genetic variation as well. Okay. Now, tomorrow, what we're going to do is we're actually going to look at a fly example and see how these 3 different things of fertilization, random assortment, and crossing over all can impact a population of flies in terms of what genes they are passing on. Okay, so that's going to be it for today. No FRQ, and I will see you all tomorrow.